Good evening, my fellow scientists. It is Tuesday, November 14th, 2017, and I want to talk about genome editing some more. Article came out in Nature Biotechnology that describes a process of using chemically modified RNA and CRISPR-Cas9, a protein, to modify a whole organism's genes. Critically, the way they got their protein CRISPR-Cas9 and the guide RNA in to accomplish this was using a non-viral vector. So instead of building all of the components that they need to accomplish their goal using a virus, which carries some risks, they built all of the components, the guide RNA, the protein, everything that was needed to build a little nanomachine to go into cells and tweak their genome. They encapsulated that into a nanoparticle and injected that in order to create the changes that they wanted. And I think that's pretty wild for a lot of reasons. One, the lengths to which they had to go to make this work is a technical feat. They had to modify their RNA strongly. They had to come up with nanoparticles capable of carrying not just the RNA, but the protein successfully and delivering it efficiently without being able to construct any of those materials in situ in the cell they're trying to modify. So their modified nucleic acid chemistry and their nanoparticle construction, both amazing. But I think that the downstream effects are going to be bigger, because if you don't have viruses, you avoid a lot of the risks associated with gene editing. Back in 10, 15, 20 years ago, there were essentially this whole idea of gene editing was really truncated, because the technologies for getting genes into cells were all viral, and they were all dangerous, and they all risked introducing mutations that you don't want along with the ones you do. Those technologies have gotten a lot better. There are alternatives, but most of these are still using some form of virus, and although they don't make the kind of systemic, nasty, mutagenic, and cancer-causing mutations that were associated with the old technology, they still carry other risks, like you can become immune to the virus that carries those genes, and that shuts down the expression, possibly causes your body to attack itself. There's other problems associated, right? So the fact that your immune system can become sensitized to the viruses that are used to correct a genetic problem raises its own host of issues. So going to a fully synthetic change means that the nanomachine that changes the DNA goes in, makes the change, and then disappears and doesn't continue to persist the way a virus would. Also great, reduces the chance of off-target effects, mutations where you don't want them, even lower. Really cool. If any of this sounds interesting, I put a link below also to the motherboard article that describes this. I think that going forward, the kinds of changes that we might see are the kinds of changes that'll make people live longer, healthier lives, and so this is good. On the flip side, there are, of course, worries that we are children playing with toys that we do not understand, and that we will find out we were playing with matches before we're done. And the unknown risks are always a bigger problem than the known risks. So I will concede that as a worry, but I'm still excited for the technology. If you like that kind of thing, tune in Monday through Friday. We talk about genome editing, biotechnology, science in general, right here in the Allen Lab.